Welcome back everyone. Now we are here with the new chapter. Okay, so what is that? This is photosynthesis in higher plants. Let us understand. First of all, we will discuss about the title first. Photosynthesis in the case of higher plants, in the sense what? I mean, basically how photosynthesis will be operated and what are their products and how their products are utilized in that. And for that, what are the requirements, how the requirements are substituted, and what are the products exactly what they are coming from that. So this we can discuss, and what factors are also affecting this photosynthesis in higher plants. All these things let us discuss in this case. Okay, right now so in your previous class or otherwise in your lower classes onwards, you are discussing about photosynthesis normally from upper primary school level onwards. So basic idea you will be having some, at least a basic idea about photosynthesis, where it is occurring, what for what purpose it is occurring, right? Anyway, we will discuss here. So in the case of introduction, what exactly mean by? Let us understand here. Photosynthesis, which is an anabolic process and moreover basically occurs in green parts of plant. Why only in the case of green parts? Because they contain something special molecules there, so that's why they will be happening there. Now apart from that, for what purpose? Otherwise, for why exactly this photosynthesis is needed, and what are the products it is getting from that? Basically, for food, ultimately is by plants only. So, the nutrition, based on the nutrition modes, there are different kinds of organisms are there. Herbivorous, carnivorous, right? Omnivores, like that. But ultimately, here food is provided by plants only. I mean, even in the case of carnivores also, indirectly food is coming from plants only. Why? Because the carnivores which are eating animals, those are again from where? Those are eating initially plants, and that plant eating animals only are eaten by again carnivorous organisms. So food is transferring, I mean the energy is transferring from level to level. So this can be studied in the case of pyramids, there we can see. Okay, now next, food. So produced by plants initially. So a kind of metabolic activity which provides food for all kinds of living organisms, either directly or indirectly. Now apart from this, all living organisms, right, all living organisms will get food irrespective of the size, shape and mode of nutrition. Now, and from actually initially how plants are doing this? How can plants can do this? By light. Which light it is? Sunlight. So initially sunlight, light energy is transforming into chemical energy in the form of food. Why? Food is nothing but a type of chemicals, right? So here, though that particular thing, it is transformed in such a way, and we are taking it as a food. Now without sunlight this cannot be happen. Definitely there should be sunlight, and moreover supply of oxygen. Supply of oxygen also one more important aspect in the case of photosynthesis. Why? Because without plants, can we imagine our life? No, not at all, right? Now for all living organisms, next, a kind of deformity is maintained by plants only. Temperature control will be there, and moreover as plants are producing, so hence they are also called as producers. Okay, producers of ecosystem. Just imagine if producers, I mean plants are not there, complete ecosystem will be in danger. So that's why development of plants and let the plants to live more and let the plants to grow more is a compulsory need. Definitely we have to promote plant growth. So then only life can be possible, not only for human beings, all kinds of biota. Either it may be some other plants starting from and towards small organisms to higher level organisms, everywhere definitely this must be used, otherwise life will be put in danger. So basically photosynthesis, where it is occurring, in green parts of plants it is occurring, and for what purpose it is going, it is providing food initially. Okay, so produced by plants. Why? Because as we discussed here, occurs in green plants and all living organisms. This food is for all kinds of living organisms, irrespective of herbivores or carnivores or whatever it may be. Now major thing what it is needed is sunlight, compulsory. Without sunlight it cannot be. And next one, supply of oxygen is also one of the major aspect in the case of photosynthesis. This is also happening. So that's why they are called as producers of ecosystem. Plants are considered to be. Right? So this is a little introduction about. So a series of experiments are there which we have to be discussed here. I mean what the person has been done, how they concluded, and what they are used for this type of conclusion. So this might be discussing here. Okay. Now Moll's half-leaf experiment. So before to that, some variegated leaf experiment is also present. That may not be, if possible, we will discuss that also. Okay, right here. Initially, what they have done here, look at it. I think in your lower classes also you might have done this. Okay, so initially they placed, they took a conical flask and placed some cotton balls which are dipped in KOH. Okay, and they adjusted a leaf half outside, half inside. Okay, half outside and half inside. Still it is attached to the plant only. Why? Because that we needed compulsory. Now they left it in the sunlight, right? So exposed to light for nearly 8 hours, 6 to 8 hours is needed. After that what happens? The leaf has been taken off from the plant. Now they tested it for starch presence or not. If the starch is present or not, they have tested by what? By giving iodine. Iodine reacts with starch and gives a characteristic color purple, otherwise blue or black, bluish black. So these all are considered. Purple colored or bluish black colored will be obtained. Now so if this color is formed, then it is confirmed that starch is present. Now they have been chosen this leaf, but only in the half of the area only they have been found that it is turned into purple, otherwise bluish black, whatever it may be. Now why? Because, sorry, not here, the other side. The upper side, which is outside the cork, I mean outside the flask, which is present. So there exactly it turned into this purple color. Now why? Because as this part is inside the conical flask, and moreover light is incident on it, right? Even though it does not produce any starch, because the KOH which is present here, it absorbed the CO2. It absorbed CO2. Why? Because CO2 is one of the chief component for production of carbohydrates, nothing but that starch. So it is not produced. So hence what happens, the chloroplasts which are present in this area, they did not receive any kind of this, like they did not receive, carbon dioxide they don't have for preparation of carbohydrates, such that what happens, ultimately there is no any production. But here the leaf outer side which is present, that have production. Okay, so that is what here. Moles half leaf experiment, leaf enclosed in a test tube containing some KOH soaked cotton balls. You can put directly KOH also here in a liquid form, right? Right. So the main property of this is it absorbs the CO2. That is the main property of this. Now the half is exposed to air. So setup is then placed into light for some time. On testing for starch, the two halves of the leaf, you must have found that the exposed part of the leaf tested positive for starch, whereas while the portion that was in the tube tested negative. That's what I have told you. So in the negative which is present, it is tested to be negative. I mean negative in the sense it is not produced anything. Why? Because we have performed some test, right? I mean applied iodine onto this leaf. So when we applied iodine onto this leaf, half of the quantity is turned into that color and remaining half is not turned into another color. Okay, so this is what happened. Now this showed the CO2 is required for photosynthesis. Yes, it proved, hence proved that CO2 is compulsory for the production of this starch, I mean carbohydrate production compulsory. Okay, right. So this is the way how they have been tested. Okay, maintaining a cork over here. So as moles, his name is moles, as he done it, that's why half leaf experiment, moles half leaf experiment has been done. I hope it is clear for you. Right. Next one, Joseph Priestley. So one of the good discovery which has been done by Joseph Priestley, one more scientist who came up with evolution oxygen, discovery of oxygen here. Okay. Remember this, Priestley discovered oxygen. Okay, no need of about the time 1774. If possible you want to remember, you can. Okay, but remember this compulsory. Right. Now what his contribution is, let us see here this picture. He took a bell jar initially, bell jar in the sense bell shaped jar. Okay, so first what he placed a candle, right? It lit up for some time. He lit the candle and kept under the bell jar. So what happened here, it lit for some time and after that it put off. So it don't have any proper conclusion on this. Then what he done, again he chose a rat, right? He chose a rat and he placed under this particular. After some time it also got faint initially. So he found that this candle light and this rat both are doing something, I mean they are removing something from that air which is required for living. So he made a small hypothesis there. Initially he made small hypothesis there and he found to know what is happening exactly. Next what he done, in the same place again he lit up the candle, again he chose a mouse and he kept a mint plant. I think you know mint, right? Right. Stolen it is. Okay. So this part, when it is placed, this both the things, I mean candle and this one, So both are initially they are safe. Okay, they are safe. So what had he made some conclusion, initial conclusion that yes, what this candle and rat is removing from the air, it is restored by the mint plant. So this is how exactly Priestley has been proved or concluded about this thing, right, about that oxygen. So later it is named as oxygen, but initially he confirmed that yes, something is present in the air which has been compulsory for the sake of life. So he performed a series of experiments that revealed that essential role of air in the growth. Now moving to here, so growth of green plants. Priestley observed that candle burning in a closed space, a bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in the closed space. Why? Because oxygen, as it is using the oxygen for, why candle will use? Candle won't breathe, right? Combustion process, compulsory oxygen is required. Without oxygen, there will be no fire. Understand? So this is why you might have seen that the fire station, the people, carbon dioxide powder will be sprayed, right? Why? Because in carbon dioxide is a bad conductor of fire, so it won't allow. Oxygen will allow. Next one. So anyway, we know that here rat anyway it will breathe in oxygen and breathe out CO2, and that we know. So that's why this is the condition. So as this is the condition, how he found? So he concluded that both burning candle or the animal that breathe in air somehow damage the air, but when he placed a mint plant, same bell jar found that mouse stayed alive and candle continued to burn. Why? Because it is supplying oxygen. Why it is supplying oxygen? Because photosynthesis is taking place in that. Continuously oxygen is released and that oxygen is used by both candle and mouse, right? Now Priestley hypothesis, plants restore to the air whatever breathing animals and burning candles remove. Okay, plants are every day restoring something that is nothing but oxygen. Now we came to know that it is oxygen, right? So this is what. Next, one more scientist, Ingenhouse, Jan Ingenhouse, okay, Ingenhouse. So he showed that sunlight is essential to the plant's process, somehow purifies the air fouled by burning candles or breathing animals. I mean, he proved that sunlight is essential. Previously what Priestley has done, he told that plants is replacing the lost to something from the air, that is oxygen. But for plants to restore it, it requires something. What is that? That is sunlight. Okay. So he had some experiment with aquatic plants. So what he showed, in bright light, small bubbles were formed around the green parts, while in the dark they did not. Okay. You might have came across this hydrilla plant experiment. Okay. Right. It was hydrilla plant placed in water. Okay. So it is placed under the water. So initially it is placed in water. Right. So here, and it is placed in sunlight as usual. Why? Because sunlight, what he want to prove is sunlight. So that's why he placed in sunlight. And after some time what happens? Slowly bubbles are coming. Why? Because here also water is there. By using photosynthesis process, small bubbles are coming, air bubbles. When air bubbles comes out, when there is some kind of gas, then only air bubbles will come out. Okay, right? Now you might have tested with this some kind of gas releasing agent. For example, if there is any leakage in cylinder, LPG cylinder what we use in kitchens, if you thinking that it is leaking, just on the nozzle you try to pour some water, if bubbles are coming out, yes, there is a leakage. So this is what happening here. Some bubbles are coming out continuously and they are escaping into this, okay, escaping into the outer air. So he had this conclusion, yes. So later he identified these bubbles to be those of oxygen, okay, ensured that that is only the green part of the plant that could release oxygen, okay. So why only green part of the plant is releasing here? Yes, there is some kind of reason for that, only green parts gland, because we will come to know anyway, chloroplast is present, so that contain chlorophyll, so there exactly this light reaction takes place and finally the process will be moving on, okay. So this is how his experiment showed that sunlight is essential for Ingen house, your wish. Right, next person, Julius von Sachs. Now what is his contribution, what he had done? Let us understand here. Sachs provided evidence for the production of glucose during the growth of the plants. So he has his glucose, okay. Now there are some experiments again here. Later studies showed that green substance in plants, I mean chlorophyll, now we are calling it as a, but by the time it is not known as chlorophyll, okay, located in chloroplast. He found that green parts in plants where glucose is made and the glucose is usually stored as starch. So some series of experiments will be done. After doing all this, they have been confirmed to be that, so this is making such a way, I mean these are made to be available for the plants and available by the plants, okay, sorry. So as we know, have some primary idea that chloroplast will possess chlorophyll, as chlorophyll pigments will take, I mean absorb the light energy, and this light energy is converted to chemical energy. Why? Because there are two phases of reactions, right? Now that will be produced here. So this is how glucose, and glucose, what it is made here, glucose is a monomer, monosaccharide, whereas starch is a polysaccharide, it's a polymer. Nothing but number of glucose molecules which are produced in the plants, so they are getting connected each other, okay, making a bond. What bond it is? Glycosidic bond. So by this bond they are forming into a large molecule. Some n number of glucose molecules will bind in this form, I mean they will make a bond and finally they will be happen to occur here. So this is what generally happens. Next, Engelmann, or don't call it Engelmann, Engelmann. Okay, so this is one of the good experiment actually, right? To put the chlorophyll using prism, split light into its spectral components and illuminated a green algae cladophora. So I think everyone is having an idea about cladophora, okay, algae placed in suspension of aerobic bacteria. So let us first go for small picture over here. Right. So in the suspension, a bacterial culture is there, including with water. Some bacterial culture is there. Now here in that he placed this cladophora, right? Now what he had done, he took a prism. I think everyone have a idea about prism, I am expecting, right? then he passed or illuminated a light. What do prism do here? It try to scatter the light like this. So what he observed that bacteria were used to detect the sites of oxygen evolution. Why? Because it is an aerobic bacteria. They require oxygen for their respiration process. Now, where exactly blue color and red color happen to present? So there, this bacteria is accumulating here. Now, why he chose bacteria? Because to indicate, where is the accumulation he want to see. Why? Because anyway algae is producing its own oxygen, so it won't move here and there for oxygen, and bacteria only needed, it is not it is not producing anything, so it only will move to the areas where oxygen is available, right? And there should be need one oxygen producing organism, that is cladophora, what he has been chosen, right? Now here where exactly it is incidenting blue and red light, remember this point, blue and red light. So for that, why? Because for blue and red the absorption will be at maximum, the light absorption for or light incident for the blue and red light is high, absorption maximum. Now the bacteria accumulated mainly in the region blue and red light of the split spectrum. So he tried to pass this light into this. Why? Because glass chamber, everywhere glass only, so lights can pass through. So he described. So the first action spectrum of photosynthesis was described. It resembles absorption spectra of chlorophyll A and chlorophyll B. So why? Because two major pigments in the case of photosynthesis, where chlorophyll A and chlorophyll B, which actively participate in the photosynthesis process, and that's why this part. Clear? So I hope this understood. So now CO2 and water giving CH2O, which is a empirical formula of carbohydrates. Okay, in a general term, general way to discuss or to tell about carbohydrates, it is written like CH2O and oxygen is released. Okay, so for this process compulsory light is needed, right? Here what is happening here, initially splitting of water molecule takes place, and that oxygen only will moving into air. This is happening here. Okay, so that is about the experiment which is done by Engelmann, right? Next, moving to Cornelius Van Niel. So usually called as Van Niel with this name, Van Niel experiment, right? So is a microbiologist who is studying about microbes and their culture or some kind of research about that. Based on his studies of purple and green bacteria, demonstrated that photosynthesis essentially light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. Okay, I will explain you. Right, now what is his initial understanding about this? When light is incident onto this particular setup, photosynthesis setup, what is happening there here? Here hydrogen source, what hydrogen source he is giving? Why? Because hydrogen is required compulsory. Why? Because in the previous one also we have seen that, you can see here CO2 is getting reduced, it is hydrogens are going to be bonding it, so that's why it is becoming CH2O. So reduction is taking place here initially. So now, so 2H2O plus CO2 gives separated two molecules of A, whatever it may be, and finally carbohydrates is produced, and one water molecule is also released, right? Why? Because in photosynthesis water molecules are also released, right? Now in this category, let us go for here. Reaction in which hydrogen from a suitable oxidizable compound reduces carbon. I mean, as it is releasing, okay, before it is normally in example mode it is showed at 2H2O, but actually what it is there, it is showed 2H2A, but generally it is water molecules 2H2O, okay, right? Now is it an oxidizable compound in the sense as it is releasing oxygen and hydrogen separately, so that's why oxidizable compound. Later it is reduced with the hydrogens, so this is what happening there, understand? Now 2H2O CO2, see here what is releasing out, so in this place generally oxygen will be present, right? That we will discuss. In green plants, water is the hydrogen donor and is oxidized to oxygen, okay? In green plants water is hydrogen donor and oxidized to oxygen. That's what I told you here. Some organisms do not release oxygen during photosynthesis. Yes. Why? Because in that this suitable oxidizable compound is different. Their substrate is different. Ultimately that only they will release. Here the components what they are taking here, those are different. So that's why it is happening to occur in a different product form. Now look here, when H2S hydrogen sulfide acts as a hydrogen donor for purple and green bacteria, sulfur is evolved. Yes, that's it. What we need here, I mean for photosynthesis hydrogen is needed, so that's why sulfur is releasing out. How oxygen will be released out, understand? So Neil concluded that oxygen is evolved by the green plants comes from water and not from carbon dioxide. So previously people used to believe that it is from carbon dioxide. Why? Because as they found this to be first, so that's why they believed that in such a way, okay? So where C6H12, it is not, it is subscript actually like this, C6, it is written by side, so please mind it. C6H12O6, I mean glucose nothing but represent glucose. The oxygen released in is from water. This was proved by using radioisotope techniques, O18, right? So this was used in the case of radioisotope. Why? Because they will employ this for exactly binding with them, so they have some affinity towards that, so that's why they employed this O18. And finally they found that, why? Because when employing itself they used the O18 in here, so this is only releasing out, so that's why they can conclude that yes, this is from water molecule but not from carbon dioxide. Why? Because both are having oxygen in them, H2O, CO2, so both are having oxygen in them. So there should be there should not be a confusion. So this experiment removed that confusions and finally gave us some kind of clearance. So when H2S acts as hydrogen donor for purple and sulfur bacteria, sulfur is evolved. So this is the equation for that. So 2H2S plus CO2 under the light influence, it giving carbohydrate separately and 2S. Okay, so this is what happening to occur, right? So 2S and this reaction is, I think so, incomplete. Okay, no, don't mind it. So Neil concluded that oxygen evolved by the green plants come from water and not from carbon dioxide. By this he concluded that yes, I think it is repeated, just never mind about that. Okay, so this is how. So what is the contribution of Neil here? He concluded that this oxygen which is coming from, I mean the some series of experiments which is done by remaining scientists, one person concluded that something is present in air which is useful for life. and later on one person concluded that that releasing thing, it is compulsory in the case of sunlight only, he told, and one person told that it is by plants. So like that, everyone who gave their conclusions are helpful for the mankind and even for next research studies. So here, so Neil concluded that this is oxygen is from wind. So how it is happening, how this H2O will be divided? So initially it is released out, okay. This process is called, breakdown of this water molecule is called as photolysis. First option will be hydrolysis. So in a hurry we used to believe that hydrolysis, oh, this is water, so ultimately we will put that as simple. So please don't do that. It is photolysis. Why? Because light mediated breakdown of this water molecule, that's why photolysis, okay. So this making them separate. Clear? So I hope this is done, and this is his contribution. Next one, he is also same, Engelmann. So he proposed that oxygen evolution, exactly when light is initiating, and especially at blue and red light. So here the bacteria is accumulating there. Why? Because availability of oxygen is more there, so that's why they are obtaining there. So in the action spectrum, light spectrum, we can see different peaks, so at different lights. So by that we can observe, we can understand where exactly absorption will be the maximum, right. Next, remember this blue and red in the case of this Engelmann's experiment, okay. So it resembles the spectra of chlorophyll A and chlorophyll B. You remember this also. Now Sachs provided evidence for glucose. So his contribution is glucose. Glucose obtained from this. So that is his next one. Ingenhouse. So he is proving that some aquatic plants, he showed this, in bright sunlight small bubbles are coming out. So those bubbles are nothing but oxygen only, okay. A gas is released, so that could maybe oxygen. He gave some initial confirmation. So next, in the case of this Priestley, so one of the good prominent person who discovered oxygen, right. So this is how this experiment has been gone, and used to this. So next, Moll's half-leaf experiment. So this, which continued that yes, starch is synthesized in the leaves. Yes, come back to the introduction part here. So basically photosynthesis takes place in the case of green parts of the plant only, as we all know this. Now for what purpose? Initially it is green plants are autotrophic. Initially they produce food for themselves, and remaining all biota, all kinds of organisms, they will produce the food, okay, right. So among that herbivorous, not only herbivorous, even carnivorous also indirectly dependent upon plants for this. So for this production of food, for them compulsory sunlight is required. Without sunlight this cannot be done. And not only by that, photosynthesis, supply of oxygen also will be present for all kinds of living organisms. So as they are providing food for all kinds of living organisms, it is also considered to be producers of the ecosystem. So compulsory we can expect two questions minimum from this photosynthesis lesson. So we should be very careful in this case, right. Now this is only introduction part and what we have completed. This is only introduction part and what we have completed here, but remaining parts in the case where we can discuss about the light and light reactions, dark reactions also, and about the photosystems. So majorly where we can get some marks from that. So right, so when we came back, we will discuss with that remaining parts, right?